Hello everyone, my name is Wisdom Akurugu. The focus of my poster presentation is on SNP rs5744934, of which the CC genotype protects asthmatic children on corticosteroid treatment against hypothalamic pituitary adrenal suppression. Inhaled corticosteroids are recommended medications for the treatment and management of asthma. However, these can induce hypothalamic pituitary adrenal axis suppression, HPAS. When used over long periods of time, can lead to adrenal destruction. The clinical features of chronic HPAS are non-specific and can be misinterpreted. 16% of asthmatic children on ICS develop biochemical HPAS, and there is no single biochemical parameter that can be used to assess HPAS. The aim of this study, therefore, is to identify genetic variations that may predict susceptibility or protection against HPAS. This is a cross-sectional study involving 96 children who underwent an overnight miterapone test and their clinical samples collected, including some covariates. DNA was extracted from the salivary samples and sequenced on the ion torrent platform, and then processed with various software tools. HPAS was defined by cortisol less than 83 nanomoles per milliliter, or all these other variables cut off. With these definitions, association and regression analysis were conducted with the Plink and R softwares. Significant SNPs were further prioritized using the functional mapping and annotation tool. 11 significantly associated SNPs with HPAS were identified in the genomic locus on chromosome 12. 2 of these lost significance after regression and adaptive permutation. 3 were significant for the dominant model, 4 were significant for recessive, and 4 had inconclusive results. The association summaries for the significant ones are shown on this table. SNPs that were significant under the dominant model are indicated as red, and those under the recessive model are green. My focal SNP, the focal SNP of this poster presentation, was found to be recessive. Further analysis showed that the focal SNP was inversely associated with HPAS defined by these 3 variables, and linearly also associated with postmiterapone ACTH values. The most striking finding was that no homozygote variant individual was suppressed. The box plot here shows that the mean square root transform postmiterapone for the homozygous variants is significantly higher than that for both the heterozygous and wild type. Plot A provides detailed information about the locus of my focal SNP. All genes mapped in this locus are colored red on this first row. The focal SNP is mapped directly to the exonic region of the DNA polymerase epsilon catalytic subunit gene. The other rows provide information on the deleteriousness score, the regulon DB score, and expressing quantitative trait loci within this locus identified. My focal SNP had a deleteriousness score of 23.3 and mapped to zinc finger genes plus repeat chromatin state. All the SNPs except the focal SNPs are expressing quantitative trait loci in the adrenal and pituitary glands on these two genes. Within the locus, the other 10 significant SNPs were not in strong LD with the focal SNP. No significant interaction was observed among the 11 SNPs in terms of effect on postmetabolite ACTH. The observed protection is therefore attributable to the focal SNP within the locus. The SNP also results in a change from medium size and polar asparagine to small size serine amino acids. This change may be enhancing the catalytic activity of the subunit. In conclusion, the variant allele of this focal SNP is associated with high postmetabolite ACTH. The CC genotype protects asthmatic children against hypothalamic and pituitary suppression. If this is confirmed in a larger sample and validated in a different population, this SNP would be a genetic marker for screening HPAS. I'm grateful to the South African Thoracic Society and the National Research Foundation for supporting this research, and thanks to the H3A Bionet for sponsoring my participation in this conference. Thank you.